Assalamualaikum and hello to all fellow judge and my friends. My name is Siti Noliana binti Daud from Faculty of Fisheries and Food Science. Today, I want to present about my final year project. The project title is Species Composition and Diversity of Mangrove Macroalgae from Avicennia Habitats at Setiu Wetland Terengganu. Problem description. Background of study. The mangrove swamp plays many roles in marine environments, not only for the benefit of humans, but for the plants and animals that live in this ecosystem. Mangrove areas are natural nutrient filters and recyclers. It also provides underwater habitat and offers critical nursing environment for juvenile or fish species. Macroalgae associate with mangrove may provide carbon to primary consumers in aquatic food web. This is because the mangrove microalgae usually found growing epiphytically on pneumatophores, roots, stems, sediments, and other substrates. Next is statement of problem. Studies on the composition of mangrove microalgae species in Malaysian waters are limited. There are only a few scientific information about mangrove microalgae in Malaysian water. Moreover, studies on the diversity and composition of mangrove microalgae in Avicennia habitat lagoon ecosystem are also limited. The ecological factors contributing on the distribution and abundance of macroalgae, especially in Avicennia habitat, is not focused well and receive little attention. Next is significance of study. The finding of this study was provided baseline information of mangrove microalgae in Avicennia habitat in a lagoon ecosystem. The suitable ecology of mangrove microalgae also have been known. Then, the suitable mangrove could be a potential candidate for aquaculture industry and biotechnology development. These findings can also help to develop strategies to promote better rehabilitation of degraded mangrove habitats and ensure these unique ecosystems survive and flourish. Objective: observe the diversity and composition of mangrove microalgae in Avicennia habitat in Setiu Wetland Terengganu. Purpose approach. Study area. This is my location of study area in Setiu Lagoon Terengganu. Sampling of microalgae. The specimen was collected from Avicennia habitat from Setiu during the low tide. Pneumatophores of mangrove was randomly harvested during course of study. Pneumatophores was cut with cutter and preserved in formalin. Then it will be kept in a Ziploc plastic bag. The specimen immediately was transported to UMT laboratory inside of ice box and transferred it to the chiller in 4 degrees Celsius for further study. Then the morphology and taxonomy identification of microalgae species. Data collection in situ hydrological factors such as pH, temperature, salinity, turbidity, and dissolved oxygen. Laboratory analysis. The length and radius of pneumatophores will be measured by the caliper. The surface area of pneumatophore will be calculated as pi dh, where pi is constant, d is radius of pneumatophores, h is length of the pneumatophores. Result: the microalgae species diversity and composition. 3 species of microalgae, Caragosa agasawarensis, Chaetomorpha minima, and Bostorchia anomala, were recorded growing on pneumatophore from Avicennia in this estuarine mangrove habitat. The macroalgae species Chaetomorpha minima contribute the highest species density for basal area compared to apex segment. Microalgae species number of Caragosa agasawarensis and Chaetomorpha minima were found significantly different between the apex and basal part of Avicennia pneumatophores, while Boswellia anomala was not significantly different. Discussion. Generally, Boswellia and Colocosa are common species that found in mangrove ecosystem. Assemblage composition of mangrove ecosystem into tropical coast. The Boswellia anomala was rarely reported species of mangrove microalgae in Malaysian water. The microalgae species Chaetamorpha minima contribute the highest species density for basal and apex than other species in pneumatophores from Avicennia habitats in situ wetland. The highest species of this mangrove microalgae was found to be influenced by nutrient availability, temperature, and light intensity. The water temperature at the surface of water body is physical factor influencing microalgae growth, abundance, and geographical distribution in different regions. The higher number of air and water are also suitable light intensity can support microalgae growth and result in higher species diversity and composition. Conclusion. There are three species that were found from this study, namely Boswellia anomala, Chaetamorpha minima, and Caloglossa agassizowarensis in this mangrove ecosystem. The higher distributional record of mangrove microalgae in situ wetland Terengganu was for the species Chaetamorpha minima. The microalgae distribution in these wetlands habitats probably be influenced by some abiotic and biotic factors such as water nutrients, dissolved solids, and salinity. That's all from me. Thank you for watching.